So, well, saying the usual thing that it's a great pleasure to present Alberto is too little. It's an extraordinary pleasure to present Alberto, to whom I know now for 42 years, Alberto.
42 years. 42 years. Well, no, we met before, maybe 43.
No,
43.
we met in 1980.
Okay.
We met because I was giving a recombinant DNA course in Argentina. Alberto was finishing his PhD and already was really very visible the capacity of Alberto. So I immediately proposed to him to come with me in England. At that time I was in Cambridge, but then we moved to Oxford and Alberto joined me in Oxford. He has, well, at that time the lab was very productive and Alberto was a key figure there. I would say that we almost nearly have been the first to see alternative splicing. Sure, we were the first to reproduce it in vitro in the mini gene. And well, I am not going to do a very long story. I could. I have written 2 pages to what I want to say.
Please, no, please.
But Alberto, let's take it from Alberto when he returned to Argentina. He continued to produce discoveries, particularly the relationship between the speed of transcription and the alternative splicing. Alberto was always sort of a hard defender of basic science, uncompromising. And that at times cost him a bit of trouble with the ministry that was very, how could I say, very application and translation. But to demonstrate that basic science is very important and that one has to keep doing it, because from there is when the great things come out. Alberto, doing these things very diversified in plants, in animals, how demonstrating the importance of the speed of transcription, what was determining it, and doing this, I arrived to a point where this knowledge was inserted in a study of a disease called SMA. And even, I don't know, have you managed to get, well, he will tell us now.
You mean clinical trials? You mean clinical trials?
Yeah.
No, not yet.
Because there was an antisense therapy for SMA, but actually that was already improved, modulating the speed of transcription. And then some molecules have shown an effect on that. I think that Alberto will tell us something of that today. And so I leave the only thing I want to say, he was my best collaborator ever and my best postdoc. And I am very fond of him. Alberto?
Well, thank you, Tito. Actually, I keep writing that you are my mentor, my biggest mentor. So, and I still, when I talk to my students, to my graduate students or postdocs, I still quote you many, many times because you taught me the basis of molecular biology in a broad way, and also the importance of the integrity in science. that I had some information about it, but it was all confirmed with you. So I thank you for this invitation, for the introduction, and for having been a light in my career and my life.
Thank you.
So good morning, everybody. We are here at 10 a.m. It's in Buenos Aires. It's very, very hot. So, but I don't have air conditioning in this room. This is at my home because I didn't want to go to the Institute just in case the connection failed. So I'm going to share my presentation. Is it okay now?
Yes, it is.
Yeah. So I cannot see you and I cannot see my face, but I guess it doesn't matter. You see me.
We can.
Okay. So as Tito mentioned, I'm going to talk about the control of alternative splicing by mainly Pol2 transcription elongation, and how this knowledge late in the last years allowed us to conceive a combined treatment for spinal muscular atrophy. So alternative splicing, as Tito mentioned, at the beginning was more conceived as an exception, an interesting exception, but now we know it's a rule. It's estimated to affect the expression of nearly 95% of human genes. Some of it is noise, but not all of it. Most of it is meaningful. It explains how a vast protein diversity can be achieved with a limited number of genes. And the importance of alternative splicing is that mutations that affect alternative splicing, either the regulatory sequences or the actual sequences of the alternative event, are a widespread source for human disease. And in my lab, we're studying alternative splicing regulation that not only depends on the interaction of splicing factors with their target sequences in the pre-mRNA, but is coupled to RNA polymerase II transcription. So this is how I explain alternative splicing for a lay audience. You are not a lay audience, but actually this is the metaphor that if pre-mRNA is like a fabric cut, you know, the spliceosome will cut this fabric in different ways, and it will depend where it cuts and where it saws, that it can produce, for instance, two different variants of the same jacket, a single-breasted or a double-breasted, and two different proteins. So the extreme case of alternative splicing is a case in Drosophila that has been described in 2000, and is the DSCAM gene, in which one gene can produce up to 38,000 mRNA variants in 4 regions of alternative splicing with mutually exclusive exons. This region has 12 alternative, this one 48, 33, and 2. So, but there are two or three things to say about this. First, it's not noise, it has a function. I don't have time to explain it, but it has a function. Second, the mechanism has been proposed of the choosing of the different variants. And the third and most important is a philosophical problem, is that this Drosophila gene has more mRNA variants than the number of genes the fly has. So the fly has 16,000 genes. So this is something we could not imagine at the time I was doing the postdoc in Tito's lab, that there were genes that were producing more variants than the actual number of genes that an organism has. So at the time we were in Tito's lab, we were studying the fibronectin gene, and we found that there were 3 regions of alternative splicing with 5 variants in this region, 2 in this one and 2 in this one, which makes 20 messengers from a single gene. So we were very proud, but actually the DSCAM example beat us. So splicing is mostly co-transcriptional. It means that it takes place before RNA polymerase reaches the end of a gene. Of course, for shorter genes, this is not strictly true, because RNA polymerase II transcribes to the end of the gene and then splicing occurs. But in most long genes, that this one in Drosophila, you can see here an EM picture with that I made the cartoon here with the DNA in black, the nascent pre-mRNAs in purple. This is the direction of transcription. The star is the promoter. And you see that before all these black dots that are RNA polymerase II complexes reach the end of the gene, the pre-mRNAs are being spliced. This is a spliceosome, and these are loops that are produced during the splicing process. So splicing is co-transcriptional, and the fact is that co-transcriptionality doesn't necessarily mean coupling between the transcription machinery and the splicing machinery. When we talk about coupling, when there is influence of the transcription machinery on the splicing event, or sometimes vice versa, the splicing event affects the transcription machinery. So we were studying this coupling for years, and essentially there were two modes of coupling that are not mutually exclusive. There could be changes in Pol2 elongation rate, what we call kinetic coupling, and or changes in the recruitment of processing factors to the RNA polymerase II itself, to its carboxyl terminal domain, to the chromatin marks, or to nascent RNA, what we call recruitment coupling. But you will see that these two mechanisms are really interacting. So I will, my talk will be focused on changes in Pol2 elongation rate, on the kinetic coupling, I'm going to talk about the role of transcriptional elongation in alternative splicing, what are the effects of chromatin structure, and how this knowledge was finally applied to a possible combined therapy of spinal muscular atrophy. So we found that at the beginning of our studies that slow elongation produced higher exon inclusion. So the model that we were in a way proving was the first come, first served, in a way that if this is a gene with an alternative exon and has a weak 3' splice site that is probably competing with a strong 3' splice site, when transcription is at a certain speed, then the pre-mRNA is presented to the spliceosome components, and the stronger side outcompetes the weaker side, and that would lead to exon exclusion. But if elongation is lower, then the weaker side is made first, then there is a window of opportunity for the binding of components of the spliceosome to the weak side, and then there is binding to the stronger side, so this would lead to exon inclusion. But in the fibronectin exon we were studying as a model, Manuel de la Mata found that actually the second intron is spliced first and the first intron is spliced second. So there would be inclusion, but this is not a problem of the order of intron excision, but a problem of recruitment of splicing factors to the weak side before there is recruitment to the stronger side. Is what it is called a window of opportunity, as I mentioned. So this mechanism in which slow elongation promotes higher exon inclusion applies to 50 to 80% of the elongation-sensitive alternative splicing events, which are not, of course, the majority of the alternative splicing events. Most of the alternative splicing events are not sensitive to elongation. I would say that up to 20% of the non-alternative splicing events are sensitive to elongation, and of them, between 50 and 80% follow this rule of slow elongation, higher exon inclusion. But lately, a second mechanism of the same elongation control showed that slow elongation in certain alternative exons could produce the opposite effect. It means exon skipping. So about 20 to 50% of the elongation-sensitive alternative splicing events follow this mechanism. And in my lab, a French postdoc, Gwendal Dujardin, found the mechanism for one of those examples that in which he showed that an alternative exon has a strong 3' splice site compared to the downstream one. So if transcription is fast, there is no time for an inhibitor, ETR3, to bind to the target site, and that would lead to exon inclusion. But if transcription is low, he showed that there is more time for ETR3 to bind to the target site, and then it binds together with TDP43, which is one of the proteins that Tito characterized in depth, and he opened a new field. But in our case, is the partner of the TDP43, ETR3, in which it binds to the target site, displaces the positive factors, and inhibits exon inclusion. So this second mechanism, I beg you to recall it, because this is the one we are going to exploit in the case of spinal muscular atrophy. It means that these exons in which slow elongation promotes skipping are called type 2 exons, and the previous one in which slow elongation produce higher exon inclusions are the type 1 exons. So the role of Pol2 elongation was confirmed by the use of a slow mutant of RNA polymerase II. This mutant had been characterized in Drosophila, and many years ago, together with my colleague David Bentley from Denver, Colorado, we produced the same mutation in a plasmid expressing the human large subunit of RNA polymerase II. It's a change of an arginine for a histidine that makes it slower. And the way we try to exploit, to show the importance of this genetically mutated slow polymerase is co-transfection of cells with a mini gene, as Tito mentioned. I remember the day 40 years ago or more, Tito and I went to ask Nick Proudfoot next door in Oxford how we could test alternative splicing using mini-gene transfections. And then we immediately produced a mini-gene similar to this, in which you transfect cells, you put your alternative exon in the middle of the exon of another gene, under the control of a promoter. So you can see the expression of this alternative splicing in the transfecting cells. But in this case, we co-transfected with plasmids expressing either the wild type or the slow polymerase. But these plasmids also bear a second mutation that made the protein resistant to alpha-mannitine. So if we treat the cells with alpha-mannitine, which is a drug that inhibits Pol2, normal Pol2, so the endogenous Pol2 is killed, is inhibited, and then degraded, and our mini-gene, and in fact the endogenous genes too, are transcribed only by the alpha-mannitine resistant polymerase that could be either wild type or slow. So this is just to show an example. I'm not showing the controls. This is an RT-PCR in which the top band means inclusion of the alternative exon, and the bottom band means skipping. And you can see that when transcription is performed by the slow polymerase, this fibronectin exon 33 that, well, it's called EDA in Tito's lab and ED1 in my lab, but now it's exon 33. And then inclusion goes up many times. But in the case Gwendal was studying, which is the CFTR exon 9, slow polymerase causes skipping. So actually, these are two examples of type 1 exons and type 2 exons. As I mentioned, remember that the type 2 exon is the case of the spinal muscular atrophy. So more recently, our friend and colleague Javier Cáceres in Edinburgh published a paper in EMBO Journal in which he created mice, knock-in mice with the slow polymerase. Here, he doesn't use alpha-mannitine because it could be a perturbation for general embryo development. So he did a knock-in, a very sophisticated knock-in, in which all the mice express either the single allele for the slow polymerase or the homozygous. And actually, expressing the slow polymerase, this arginine 749 histidine, is embryonic lethal. So the mice do not progress, but he could grow the embryonic stem cells and put them in culture. And he showed that there are many changes in gene expression and in alternative splicing. And most importantly, those cells in culture lose the capacity to differentiate to neuronal precursors. So there is a simple change of 3 to 4 times in the speed. Not only is embryonic lethal, but when the cells are rescued, they can only they can replicate, but they cannot differentiate into neuronal precursors. So what can change Pol2 elongation in a more physiological or pathological way? Of course, the slow mutant doesn't exist in humans. It exists in Drosophila, and it causes some phenotypes, but doesn't exist as far as we know in mammals, except for the knock-in performed by Javier. So it could be, in fact, modulation of Pol2 intrinsic activity, either via phosphorylation, its carboxy terminal domain, or promoting association to elongation factors. And this was mainly the work of Manuel Muñoz in my lab, and I'm not gonna describe this because I want to focus on the second possible mechanism, that are changes in the template chromatin structure that could limit it or facilitate elongation. So I just depicted it with a track in a dust road, in which, of course, the speed is lower than if you go on pavement. But actually, I would like to stress right now that most people who study transcription and probably know very little about splicing, they always focus on chromatin structure changes at the promoters. And we focus far away from the promoter, intragenic, inside the gene, downstream of the promoter, mostly around the alternative splicing events. So this is a very important thing, because sometimes there is an understatement that chromatin effects on transcription may only affect promoter strength, which is not actually true. So I will just summarize what we found about alternative splicing regulation by chromatin. So we found that in different cases, if the chromatin structure is compact, then Pol2 goes low, and that would affect the alternative splicing events that are embedded in this region. But if we treat the cells, for instance, with a chromatin opener with valproic acid is an inhibitor of histone deacetylases, so histones remain highly acetylated, and then chromatin is relaxed and more open, and we show that Pol2 goes faster, and if this affects a region of an alternative splicing event, then there is a change in alternative splicing. So in this case, you see clearly this is a type 1 exon, because slow makes more inclusion and fast makes more skipping. But you can imagine exactly the opposite for the type 2 exons, like the CFTR exon 9, or the one we're going to study in spinal muscular atrophy. So what is important for opening or closing of the chromatin are the histone marks, which is a whole domain that we are little experts in this, because there are plenty of people working on this, and we enter this after we found the effect. But actually, in a simplified way, if there is marks like methylation of lysine 9 of histone A3, this is a silencing mark. It promotes chromatin compaction, and that would affect alternative splicing in promoting slower elongation. But the same residue, lysine 9 of histone A3, could be acetylated, as I mentioned before, and this is a permissive mark and will open the chromatin and promote higher elongation. So these are the two marks we studied more, but also we studied K27 methylation, but it is a restrictive mark that promotes heterochromatin too. So in different cases, we propose a model in which there is alternative chromatin that could affect alternative splicing on the same gene in two different situations. For instance, in cells in culture, if you promote neuron depolarization, then there is higher histone H3K9 acetylation, faster Pol2 elongation. In the case of the NCAM gene exon 17, I think, is skipping. While if in the same gene in a different condition, like neuron differentiation, has higher kinase methylation, a more compact chromatin structure, and that would lead to exon inclusion. So with all this knowledge, why we fell into studying spinal muscular atrophy. So I think I have time to tell you the story. We never, Tito say, we never did anything related to disease, mainly because we didn't want to promise neither the authorities nor the families of diseased people anything that we couldn't give some light to the field. But about 8 years ago, the families of Argentinian spinal muscular atrophy patients, they're mostly kids, came to my lab and said, we know Adrian Krainer, who is a friend of us and who I will show his achievements, is about to get the therapy for spinal muscular atrophy approved by FDA. We know you also work in alternative splicing, so we want you to investigate, to do research on the disease here in Argentina, and we are going to support you. So at the beginning, I said, okay, thank you very much. I appreciate your offer, but I said no. I said no because I didn't have anything to improve in what it was already known. I was not an expert in spinal muscular atrophy, and I didn't want to raise any expectancies in the families, because this is very, very critical. You know, these are people who are suffering from a life that is very, very difficult for them and for their children. But then it occurred to me to do an experiment with my graduate student. Well, he wasn't a graduate student, he was an undergrad at the time, Luciano Marasco. And we wondered whether the speed of elongation affected the exon that you will see is the key for the therapy. So let me tell you first what happens with the disease. This is an autosomal recessive disease, SMA, in which the motor neurons are affected and there is poor innervation of the skeletal muscle, which leads to atrophy of the skeletal muscle. And in the most severe way of the form of the disease, if the kids are not treated at that time with mechanical ventilation and now with drugs, they would die in a few months, a few weeks or months, because their muscles cannot contract. Okay, the thoracic, I mean the ventilation, respiration muscles cannot move. So what is the cause of the disease? The disease is the absence of the SMN protein, because the SMN1 gene is mutated. So as I tell you like this would be embryonic lethal in humans, but it is not embryonic lethal because we humans have a second gene named SMN2 in the same chromosome 5 that is a recent duplication in evolution, and that it has 11 base pair difference with SMN1. We all have this gene. Both are about 30,000 base pairs long, but this has 11 changes. And among those changes, there is a base change in exon 7 that makes it skipped during expression. So in SMN2 gene produces very little of the healthy protein, and most of the exon 7 minus altered protein, 80% of it, that is useless. And it's on top of it is degraded. So Adrian found that there is a protein, an inhibitor of exon 7 inclusion that binds a specific silencer sequence in intron 7. So he developed an antisense oligonucleotide therapy, and the antisense oligo is called nusinersen, or the commercial name is Spinraza. And this is Adrian, and what this Spinraza or nusinersen antisense oligonucleotide does is that it binds to the pre-mRNA just at the target sequence of the THNRPA1 and A2 inhibitor, and it reverts the problem of exclusion of exon 7, promoting its inclusion, which makes SMN2 gene to promote 80% or even sometimes more of the healthy protein and only 20% of the incomplete protein or truncated protein that is degraded. So he, of course, did all the experiments in cells and mice and monkeys, and then it was approved for use in humans in 2016. I just want to mention here that this is not strictly speaking gene therapy, because in gene therapy, what you do is to add a healthy gene to the genome or to correct the mutation in the damaged gene. In this case, we are not touching the SMN1 gene, but waking up the correct splicing in the backup gene, in the SMN2 gene. Okay, I mean, I wait for questions at the end. If there is something, someone of you is urged to ask questions right now, I would be happy. We are in Zoom. But anyway, nusinersen or Spinraza acts by displacing a negative protein factor and producing more of the good protein by the backup gene. So I will show you a movie that is very moving. Of one of the kids that was treated during the clinical trials with Spinraza. So his name is Cameron. He was diagnosed with SMA type 1 at 5 weeks old. Doctor said he only had 6 to 12 months to live. He could live longer, postrated with mechanical ventilation, but his parents enrolled him in an experimental drug trial called nusinersen Spinraza by Ionis Pharmaceuticals, and the drug was developed by Adrian Kleiner at Cold Spring Harbor, New York. Just as a funny note, the company originally was named ISIS, but since they didn't want to be confounded with the ISIS, they changed the name to Ionis. So the video shows what happened. So this is 7 weeks old. Before starting the treatment, he was diagnosed at the genetic level. He has belly breathing, little movement, and bell-shaped legs. Now he's 4.5 months old. He's grabbing objects. He didn't die, first of all. He was treated with 2 or 3 intrathecal injections per year. And now he's 1 year old. He's holding a heavier toy. At 20 months old, he's standing. And now he was 3 years old. He was riding a tricycle. Now probably Cameron is, I guess he's 7. This is in the web anyway. It's available to everybody. Not such a short film, but all the movies the parents recorded. So you can imagine, I go with Adrian to the meetings of the SMA families in United States. And when Adrian enters a room, because the families go to the meeting too, it's a scientific meeting with the families, he's like God. I mean, he was the first to cure a neurodegenerative disease and brought happiness to many, many families. So the question we asked before, or after saying no to the Argentinian families, is whether SMN2 exon 7 alternative splicing was controlled by POL2 elongation. And the answer was yes. In cells in culture, if we transcribe the gene with the slow polymerase, exon 7 is more skipped. I mean, there is skipping. So clearly, as I told you, it's a type 2 exon. But then this was with mini genes in culture. But we went back to a published paper by David Bentley a few years before, in which he had done cells in culture with a slow and a fast polymerase, another mutant that goes faster. And so we asked him to look at the SMN2 exon 7 region, and actually we confirmed that while the slow polymerase inhibits exon 7 inclusion, the fast polymerase promotes inclusion. So the model was confirmed independently in cells from another lab, and either more confirmed by the use of a fast mutant. So we reasoned that chromatin opening should increase SMN2 exon inclusion, exon 7 inclusion, by promoting intragenic Pol-2 elongation. So we decided to use a histone deacetylase inhibitor. We used 2 or 3. I will show results with trichostatin A or valproic acid that, by promoting histone acetylation, will open chromatin. So we decided, Adrian decided, it was a collaboration with him, not to use, strictly speaking, nusinersen because of proprietary reasons. So we decided to use ASO1, which is exactly the same, has 2 more nucleotides, but Adrian had shown in many papers that they behave exactly the same, both in cells and in animals. So every time I mention ASO1, I mean nusinersen, okay, or Spinraza. So you can see here in cells in culture that in HEC293 T cells, that while ASO1 in suboptimal doses promotes exon inclusion, that TSA also promotes inclusion. You will see that this happens in cells, but not in mice. I mean, TSA or VPA have no effect per se in mice, but both have an additive effect, okay? So there is more inclusion caused by trichostatin A, which is a histone deacetylase inhibitor that inhibits class 1, class 2, and class 4 histone deacetylases. The same happened with valproic acid, which is a class 1 inhibitor, and also ASO1 promotes inclusion, VPA, valproic acid promotes inclusion, and both are additive. And we decided to continue with valproic acid because it was approved by FDA and its use in epilepsy and other convulsive neuron diseases. So I will not show you all the controls we've done in cells in culture, because after 1 year, of course, I had to say yes to the families. And after 1 year of work by Luciano Marasco, I told him you should go to Adrian's lab to inject mice. Why to inject mice there? Because we don't have such a big facility, mouse facility in our country as the one needed for this kind of experiments, and they have all set up for the SMA model. So we decided to do immediately experiment with SMA mouse model, which is a Taiwan strain. What is this strain? Well, in mice, there is only one SMN mouse gene. So when it is mutated, the 2 alleles are mutated, this is embryonic lethal. I told you that in humans, this does not happen because there is a backup gene, but the mouse has no backup gene. So the Taiwanese researchers in 2000 created a transgenic mouse with the human SMN2 transgene. So in these conditions, this is not embryonic lethal. They make a little bit of the right protein, which is enough to, they get birth, they are born, and they live for 7 days. So any treatment you do to the mice during those 7 days that improves the inclusion of exon 7 will, of course, prolong their lives, and they will gain weight and survive. So this is a fascinating model because we are testing actually the human SMN2 transgene in a mouse. So we, following Adrian's protocols, did a single subcutaneous injection of nusinersen like of ASO1 and or the HDAC inhibitor. And the injections were there subcutaneously, as I mentioned, just at day 1 or 2 after birth, and then nothing else. So all the results you're going to see are of a single injection. That doesn't mean that if you inject more, you get better results, but we were using this as a proof of principle, not as a protocol for actual treatment, okay? So these are 11 days old mice. These were injected with suboptimal doses of the ASO alone, and the heterozygous is the wild type. You see the ones injected with suboptimal doses of ASO1 are smaller, but they survive because these are 11 days old. We were talking about 7 days old. And these are the ones at the same age, of the same age, but you see that they were injected both with the ASO1 plus the histone deacetylase inhibitor, and you will see that they are bigger, and they are almost the same size as the heterozygous, which is the wild type. So now they are 40 days, 42 days old, and you see the size difference. This is a one treated with ASO1 and has other phenotypes. Among the fur is different. They have different behavior, but the ones injected with ASO1, in this case trichostatin A, are much bigger and better. So if you measure the body weight gain, and this is what I mentioned, the ones injected with VPA alone die at 7 days. So VPA does nothing. It doesn't improve the survival, okay, per se. And this is consistent with the fact that VPA had been used in a clinical trial on the basis that it would increase SMN2 gene transcription and then produce more of the healthy protein, but it failed. So the trial was called Carnival because they had to co-inject or co-administrate carnitin. But VPA has no effect in mice. So the purple curve is the heterozygous, the wild type. The blue curve is the ASO1 in suboptimal dose, and you see the effect on VPA here. That is improving the body weight gain. But of course, you won't see how many mice survive at 60 days. And this is seen in the survival probability curve, the Kaplan-Meier. And again, VPA alone has no effect. ASO1 at the dose we applied and in the timing we applied, all mice died after 60 days. And you see the impressive effect of VPA on the effect of suboptimal doses of ASO1 with a p-value very, very, very low. And the same happens with, well, I think the previous one was VPA. This is trichostatin A, exactly the same two curves. But most importantly, we measure the amount of the SMN protein in the different organs of the mice. And you can see, for instance, that in liver, the trichostatin A doesn't increase the amount of the SMN protein. The ASO1 does increase, and then the two together have a much better improvement of the amount of protein. And why we look at liver, kidney, muscle, spinal cord, brain, and heart? Because SMN is a protein that is important for the process of splicing that occurs in all cells. Of course, its absence is more critical in motor neurons, but it also affects the periphery. So our project is also, in a way, devoted to correct the right SMN expression, not only on the central nervous system, on the or in the motor neurons, but mostly in the periphery. Why in the periphery? Because Spinraza is injected intrathecally, but in the spinal fluid. So it goes mainly to the neurons. But there is some leakage, there is some clearance in the periphery tissues where the concentrations are much lower. And in those much lower concentrations, the effect of the chromatin openers could be positive. I'm going backwards. So one day, Luciano, who was doing the injections at Cold Spring Harbor, sent me by WhatsApp these movies, and I was thrilled. So he had gone to a lab who is an expert in behavior and had shown that the pups, 11-day-old pups that were injected with ASO1 alone had difficulties to surface, to writing themselves. But the same age, other pups write themselves immediately. So you can measure this, and this is the writing reflex latency, and this is the wild type. They write themselves immediately. The control, the mutant, takes 50 seconds to turn around. VPA doesn't do anything significant. ASO1 is improving, but ASO1 plus VPA is much better. And the second test he showed me is the grip strength. So he they put the pups on a rough plastic piece of plastic, and they measure the angle at which they fall. They try to climb and they fall at a certain angle. Actually, despite all my years in Oxford, I didn't know that this is, in English, is called a protractor, and I learned it here when I was old enough to know it. In Spanish, we call it transportador. I don't know in Italian, but actually, you see that the ones injected with AS01 plus VPA stand at higher angles, and you can measure this. This is the wild type. It falls at 90 degrees. The control falls at lower angles. The VPA does nothing. AS01 in this case, very little, but both together are very strong. Okay, so because we were curious enough to see what happened with the chromatin after the treatments, we discovered that the ASO affected the chromatin structure as well. In what way? So it was similar to what we had discovered many years ago. Mariano Alliot in my lab showed that if we transfect cells with siRNAs whose sequences are directed to intronic regions that are downstream of alternative exon, then there is a nuclear effect of the siRNA in which it promotes recruitment of enzymes that cause K9 methylation or K27 trimethylation, which creates a roadblock to elongation. And because it stops transiently elongation, it affects the downstream alternative splicing event. In the case of type 1 exon, it promotes inclusion. So we wonder if AS01, which is not an siRNA, is a piece of modified, heavily modified, maybe DNA, I don't know, create also roadblocks to elongation. And the answer in cells in culture is yes. AS01 promotes methylation of the gene or the histone around the gene. This methylation goes down when we treat with VPA, ASO plus VPA. And when we did POL2 chips, we see that there is a roadblock to elongation caused by AS01. And this roadblock is downstream of the target site of AS01. And when we treat with VPA, this goes down. So the model we were conceiving is that without the chromatin opener, of course, AS01 displaces HNRMPA1 and A2 and promotes exon inclusion of the exon 7, but at the same time, it causes the opposite effect by promoting K9 methylation in the chromatin and inhibiting inclusion. When we treat with the HDAC inhibitor, either TSA or VPA, we open the chromatin, and then both events go towards exon inclusion, which is what we found actually in the RT-PCRs. So a key question, and I will show the published result, but I will tell you our latest result now, is how pleiotropic is VPA? Well, on the one hand, I told you that it's accepted, it's approved by FDA and other agencies. So whatever secondary effects it has to be tested in humans to see it improves the cure of SMA without causing huge secondary effects, because it affects all the genes, of course. And you can see here that with VPA, elongation is augmented in all genes, while AS01 doesn't affect elongation. This is a meta-gene analysis. But actually, when we did RNA-seq to see gene expression, we found that although there are many changes, only about 70 genes cause a high differential expression, because this is the log 10 P of 5 that is considered to see a significant effect. Of course, AS01 does nothing in total gene expression. And when we treat it with AS01 and VPA, we also have about 100 genes above the threshold, I mean, the level of 5 log 10 P that is considered significant in bioinformatics, I would say. But you might also say, well, some of the genes can be key for secondary effects. Yet it's true. And surprisingly, although there were changes in alternative splicing, very massive changes, the significant ones, which are the reds and the blue dots, were not that high. So essentially, we conclude that there is a kinetic coupling. So that means that slow elongation can promote either exon inclusion or skipping, depending on the particular alternative splicing event. We have a model in which a single gene could have alternative chromatin configurations that will alter alternative splicing. And we were proposing a combined therapy for spinal muscular atrophy with Spinraza, ASO1, and drugs that open the chromatin, which in our hands do not have a huge pleiotropic effect. And the mechanism, which is the most, I would say, daring and interesting molecular biology part of these results, is that the Spinraza ASO1 has two opposite effects. The negative effect, which is creating methylation and then slowing elongation, is counteracted by opening the chromatin with the histone acetylase inhibitors. And before I continue, let me tell you that now what we did is to deliver histone acetylation specifically to the SMN2 exon 7 region. And how you do that? Using the dead Cas9 strategy. So we prepare a plasmid expressing a catalytically dead Cas9 fused to a protein that promotes acetylation, and then guide RNAs also expressing, plasmid expressing guide RNAs that would lead this dead Cas9 with an acetylating enzyme to different positions along the gene. And I can tell you, because I don't have the results now, but these were confirmed last week, that when we deliver the acetylating enzyme specifically to the, for instance, intron 6, you get an increase of the effects of ASO1. So we are conceiving that perhaps in the future, the combined therapy could be not using VPA, that is a drug that affects many, many genes, but delivering dead Cas9 with an acetylating enzyme to the region we want. So just to finish, Tito mentioned that they might, well, no, before that, well, we were fortunate enough to have the paper accepted in Cell, and it was out, what it says in press there, but it was last year. And then they, as they always offer you to illustrate the cover. So Luciana Giono, who now is not longer in my lab because she turned to do illustrations for papers, and she does very well. As you can see, we imagine a Calder mobile in which if histones are acetylated, exon 7, which is this white plate, is included in between the two other exons in the mature messenger, while if we have methylation of the histone, exon 7, which is the white plate, is not included. So it's an allegory for the findings we had on the chromatin modification and exon inclusion. But as this is the important people, Luciano Marasco, who finished his thesis last year, and now he's doing a postdoc in Nick Proudfoot's lab. Nick Proudfoot is like a friend for Tito's and like a cousin for me. We are like a, sorry, I meant like a brother for Tito and like a cousin for me. And we were lucky enough that during the pandemic, our lab was closed, but his lab was open because of the changes in the waves in the pandemic. So some of the genome-wide experiments were done in his lab. Of course, Adrian, who was key to this collaboration, Luciano, who did 99% of the experiments, the collaboration of Gwendal Dujardin, who was also in Nick's lab, and Rui Sousa Luis, who is a bioinformatician of the Carmo Fonseca's lab in Lisbon. So this is a very international team. And Jose Stigliano, you say that I say Stigliano, any Argentinian would say Stigliano. Jose is Italian. He's a son of an Argentinian who was educated in Italy in Rome, and he did his degree in Dundee in Scotland. And came to my lab to do his PhD, and now he's working with this dead Cas9 delivery to the gene and seeing how it improves the effects of Spinraza. So what I wanted to tell you at the end is that Tito mentioned that he also worked in plants. This is the only slide I'm going to show you. And this is the work of Micaela Godoy-Hertz and Guillermina Kubacka. And we started with Ezequiel Petrillo. We published a science paper in 2014. And now we found that light affects alternative splicing decisions in the nucleus of Arabidopsis. But the actual sensor is the chloroplast, and is the photosynthetic pathway which affects the splicing. And in the latest paper, we found that what light controls is the speed of polymerase in the nucleus. That's why Luciana Jono, who is the illustrator, conceived this taken from the web in a US road where there is a different speed limit for the night and the day, okay? So I have to thank the support for many years from our Hughes Medical Institute, and now mainly from QSMA, from the Argentinian families, and from the Lounsbury Foundation, together with the local agencies. So I thank you very much, and I'm welcome to answer your questions, if any. Thank you.